So our next biomacromolecule are carbohydrates. Again, four biomacromolecules: lipids, proteins, carbohydrates, and nucleic acids. Remember that proteins, carbohydrates, nucleic acids are all polymers. When we looked at proteins, the proteins are a polymer of what? Amino acids, right? Carbohydrates are going to be a polymer of monosaccharides when we link them together. So again, we want to remember that we have a common reaction, a dehydration synthesis, to link, in this case, monosaccharides together to make polysaccharides. We can break those polysaccharides apart with hydrolysis to go from complex to simple sugars, from the polymer of carbohydrate to the monomer of carbohydrate. So let's look at that difference between monomers and polymers in the carbohydrates. Now, carbohydrates are made up of three elements and three elements only: carbon, hydrogen, and oxygen. That's it. We don't decorate them with anything else. So they're less complicated than proteins in that respect. Now, as opposed to proteins, which have only 20 common amino acids, there's lots and lots of different sugars. Now, the sugars that we're talking about here now are monomers. They're small organic molecules. Some have 3 carbons, some have 5 carbons, some have 6 carbons. There's all different kinds. And if you become like a sugar biologist, a glycobiologist, you'll learn all these, but you don't have to know them now. All right? But we do want to note that there are these small organic molecules called monosaccharides, saccharide being sugar, mono one sugar, made of carbon, hydrogen, and oxygen exclusively. Now we're going to depict them in this what's called the linear form, but they'll also form this ring form in aqueous solution. And oftentimes when we're going to link them together into a polymer, we're going to actually use the ring form to link these monosaccharides together. So in this case we see glucose. So this is the linear form of glucose on the far left. We can see with a little bit of chemistry we can link it to itself to make this ring form. And that's going to be important because you can link that together into a couple different ways. But this idea now of starting to link monosaccharides together, we're starting to build polymers, right? So in this case, again, there's lots and lots of different sugars out there. When you put a glucose and a glucose together, you get a maltose. When you put a glucose and a fructose together, you get a sucrose. And sucrose is basically the table sugar, or the sugar that you have at home, that granulated sugar. That's mostly sucrose. So this now, we go from monosaccharides to disaccharides, di being two saccharides put together. After that, we basically go up to polysaccharides as we start to link hundreds or thousands of monosaccharides together. Now, glucose linked together in thousands of units we call starch. And this is the primary form of energy storage for plants. So carbohydrates, then, our first biological function for carbohydrates is energy storage. Glycogen is a much more branched form of starch. This is the way that animals tend to store carbohydrates. So again, we see the polymer of a carbohydrate can be used for energy storage. Okay? Starch in plants, glycogen in animals. Now, these carbohydrates, depending on how you link them together, can form their second function in biological systems, and that is structural. So we're going to take that same glucose molecule and we're going to form a ring. Now, when we form a ring, we can form 2 different kinds. We can form an alpha glucose ring or a beta glucose ring. And the only difference you see here is where that OH, that hydroxyl group, is either below the ring or above the ring, as you see in these pictures. Below on the left, above on the right. Now, if I have a bunch of beta glucose monomers versus a bunch of alpha glucose monomers, and I link them together, I get 2 very different compounds. So starch is an energy storage molecule in plants. It's a bunch of alpha glucose monomers linked together. This, you eat it and you break it down, and we'll do this in lab, and it tastes sweet on your tongue because you're breaking down the long polymers into individual glucose molecules, which you register as sweet. Now, cellulose is beta-glucose monomers linked together, and this is basically wood. This is the material that plant cell walls are made out of. You put this in your mouth, you can't touch it. You cannot break this down. It's roughage, right? So what we can see is glucose then as a structural polysaccharide is a carbohydrate serving a structural function. We link them together, we form these fibrils. These fibrils then are wrapped around cells, plant cells, forming the plant cell wall, which we'll talk about later, but ultimately this is what we think of as wood. So if you're sitting at your desk, the wooden table you may be sitting at is actually all glucose. Unfortunately, you can't just take a bite out of it and eat it, because the way those glucose molecules are linked together is inaccessible to your biochemistry. Right? So we have polysaccharides, or carbohydrates, for energy storage, as structural molecules, and finally, they're going to act as modifiers. So this slide shows you a combination of two of our biomacromolecules. Proteins in the center, there's the polypeptide chain folded back on itself to get a specific function, a specific form, and hence function. And then you see these groups on the upper right, upper left, and lower left. These are sugar molecules, carbohydrates attached to it. Oh no, we've combined the classes, right? And this is going to allow us to add more, even more diversity to the biomacromolecules that we can make. So sugars or carbohydrates are often small sugars, monosaccharides or disaccharides or small polymers, can be added to other biomacromolecules as modifiers, both to proteins and to lipids. So here we have a glycoprotein. We also, like I said, have glycolipids, and these are very much involved in cell-to-cell communication, how bacteria or virus cells attach and recognize cells per species and what have you. So this combination, this modifier component of sugars is very, very important to biology. And in fact, it has a special name. We call it glycobiology. So glycosylation is a word I want you to know. That's the verb for adding a sugar group onto a protein or a lipid. So if you say, oh, I need to glycosylate that protein, it means I need to add sugars onto it. And these are going to change their biological function. We create more combinations of more variability in our repertoire of biomacromolecules.